Nucleus. Nucleus is the most important part of the cell. It regulates and coordinates various life processes of the cell. It plays an important part in cell division. It contains factors which determine the heredity. Nucleus is a small spherical mass located somewhat in the center of the cytoplasm. It has a delicate nuclear membrane which is filled with a relatively dense nucleoplasm. In the nucleoplasm, there are certain thread-like structures called chromatin fibers. During cell division, the chromatin fibers become thick and ribbon-like. The fibers are then called chromosomes. Robert Brown in 1883 discovered a within the cell and termed it as nucleus. A German biologist, J. Hammerling, demonstrated in 1934 that the nucleus determines the characteristics of the cell and ultimately the characters of the individual. He conducted certain experiments using two species of green alga, acetabularia. So as we can see here, the nucleus is bounded by nuclear membrane, and this nuclear membrane is a double layered membrane made up of phospholipid. And there is a nucleolus, protein and ribosomal assembly, RNA structures are here. And then the chromatin fibers, nucleoplasm, and the nuclear pore. So we will do one by one. Nuclear envelope is a double membrane structure. Two membranes are unit membrane structures, that is at the center phospholipid layer, and on either side it has protein. Inner nuclear membrane is lines with the nuclear lamina, which is composed of lamin. Lamin is intermediate filamentous protein. We have learned the cytoskeleton structures made up of three elements of cytoskeleton structures: actin filament, microfilament, then microtubules, and intermediate filaments. This intermediate filament, one example is lamins. They are there on either side of the nuclear membrane. Outside the outer membrane of nuclear membrane is also lamin proteins are found, and inside also it is found. These are for giving the mechanical support to the nucleus. Space between two membranes is known as perinuclear space, that is in between the outer membrane and inner membrane of nuclear membrane. There is a space and it is known as perinuclear space. It is filled with the fluid. Outer membrane is continuous with endoplasmic reticulum. When we learned that it is continuous with the nuclear membrane. Nuclear envelope is interrupted by nuclear pore. This is the most important structure and it is not much explored structure. This nuclear pore is not a very mere pore. It is not a simple pore. It is having a protein assembly. It is known as nuclear pore complex. Now, nuclear pore is 30 nanometer to 100 nanometer wide pore. It is large complex structure, 30 times the size of a eukaryotic ribosome. And what is its function? It provides direct contact between cytoplasm and the nucleus, allow import and export of protein and RNA. Mainly tRNA and mRNAs are transported from the nucleoplasm to the cytoplasm for protein assembly, and import of proteins for the synthesis of DNA, like DNA polymerase or RNA synthesis is happening, mRNA synthesis and tRNA synthesis is happening inside the nucleoplasm. For all these processes, the nucleus need enzymes. All these enzymes are in the form of protein. These will be transported into the nucleoplasm. And what is nucleoplasm? The part of protoplasm found inside the nuclear membrane is known as nucleoplasm. It is composed of enzymes, dissolved salts, and different organic molecules. Then, nucleoplasm contains nucleolus and chromatin material. Nucleolus is the largest structure in the cell nucleus. Nucleolus is the nuclear subdomain that assembles ribosomal subunits in eukaryotic cells. Nucleolar organizer regions of chromosomes which contain the genes for pre-ribosomal RNA serve as the foundation for nucleolar structure. Nucleolus disassembles at the beginning of mitosis. Its components disperse in various parts of the cell, and reassembly takes place during telophase and early growth G1 phase. Nucleolus is the most prominent structure, and it is not having membrane. It is the site of transcription. That means the mRNA synthesis is happening in this particular region. And it is also the site of synthesis of ribosomal RNA. This mRNA and ribosomal RNA, which are synthesized here, that will get transported out of the nucleus through nuclear pore. Then the most important structure, which is controlling the transmission of characters from the parents to offspring, or the controlling the characters expression, protein synthesis in our body, or gene expression, that is chromatin. These are thread-like structures made up of strands of DNA and the histone proteins. The chromatin condenses into individual rod-like structures called chromosomes during cell division. The construction on the chromosome is known as centromere. At this construction, two arms of the chromosome are held together. This holds the chromatids together. Kinetochores are the disc-shaped structures on the sides of the centromere. Kinetochores are protein and DNA assembly. The chromosomes become attached to the spindle fibers by their kinetochores. The kinetochores will have proteins, and then spindle fibers are also made up of protein. So this protein-protein interaction will make sure that this chromosomes are attached to the spindle fibers for separation of this chromatids at during anaphase stage. And depending upon the position of centromere, the chromosomes are divided into four types. When the centromere is at the center of the this chromosome arm, it is known as metacentric. If it is just above the center, it is known as submetacentric chromosome. And here it is towards one side of the chromosome, and it is known as acrocentric. And if it is towards the end, if it is at the end, the centromere is placed, this particular chromosome is known as telocentric. And this arrangement will be this formation. Two chromatids are attached to the centromere. This you will find only during cell division stage. Other than that, if the cell is not at the dividing stage, then the chromosomes will appear like this, right?